Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. In this occasion, at the first Kuningan International Conference, I would like to present my research result with the title Toxicity and GCMS Analysis of Cassia Ciamea.Lamk ethanol extract. My name is Harunatanti, and my team research are Mr. Aryusmar, Mr. Bustanusalam, and Miss Sharifah Diana Permai. I am a lecturer at Binus University, same with Mr. Aryusmar and Miss Sharifah Diana Permai. But for Mr. Bustanusalam is from Biotechnology, LIPI Cibinong. Well, there are three reasons why I interested to research about johar. First, johar plants can grow in all regions of Indonesia, so you can easy to find this plant in Indonesia. The second one, the leaves, stem, and roots of johar are used by community as a traditional medicine, treating malaria, diabetes, diarrhea, etc. It's a report by Kamagate at 2014. And the third one, that's at Cassia Ciamea.Lamk, it's content of the secondary bioactivity compounds like alkaloid, tannin, saponin, hormone, flavonoid, carbohydrate, protein, steroid, terpenoid, and cardiac glycosides. It's reported by Verachev and 2011. This is the picture of Cassia Ciamea.Lamk or johar trees. The people widely planted on roadside or in the gardens. The trees with the hard stems, yellow flowers, green like Leguminous family. Classification of johar: kingdom, plantae, subkingdom, class of superdivisi Spermatophyta, divisi Magnoliophyta, class Magnoliopsida, subclass Rosidae, ordo Fabaceae, family Fabaceae, genus Cassia, and species Cassia Ciamea.Lamk. There are two research purposes. The first, testing the toxicity of johar leaves ethanol extract with the brine shrimp lethality test or BSLC method. Why I use this method? Because the research use this method cheaply, quickly, and easily. The second one, to determine the compounds in the leaves of Cassia Ciamea.Lamk leaves using the gas chromatography mass spectroscopy or GCMS method. Well, for sampling, I taken leaves from Putat village, Sindang Laut district, Cirebon Regency, West Java, Indonesia, on April 2019. The location is nearby Kuningan. The second one, the fresh leaves are washed several times with water to remove impurities at room temperature for 3 weeks without sunlight, and melt into powder using a food blender. So for this experiment, I used the powder of Cassia Ciamea.Lamk. It is dry Cassia Ciamea.Lamk leaves after I put in the room temperature for 3 weeks without sunlight, and before I make powder. Well, I use 454.99 grams of powder Cassia Ciamea.Lamk, and then I extract it by maceration technique using 96% ethanol. The sample I divide for 5 bottles, and each bottle I use ethanol. Then I leave it about 1 days or from 24 hours. After that, I filter all the sample and then replicate it 2 times for maceration, and the end all the combine. Okay, all the filtrate from maceration I evaporated at 40 degrees Celsius with rotary evaporator. It's produced about 15.9 gram extract, black extract. The black extract of ethanol, and then I fractionate it using N-hexane. It's replicated about 3 times. Yeah. After that, I separated the hexane and the water fraction. After that, the water fraction or residue was fractionated again ethyl acetate, same 3 times, and then all the fraction of hexane and ethyl acetate extract are evaporated with okay. For toxicity test with the SLC method, I use artesia salina leach grease with a weight of 50 mg and put it in a beaker containing 500 ml of filtered seawater, and then an aerosol was installed. Leave for 2 days or about 48 hours with CL lighting so that it hatches perfectly. And then I preparation of 2000 ppm ethanol extract solution by I dissolve from 40 mg ethanol extract in 20 ml of seawater, and I add 1% DMSO or about 5 drops to increase solubility. Okay, toxicity test with the SLC method use 5 kind of solution. So I mix another solution with concentration 100 ppm, 50 ppm, 20 ppm, and 10 ppm. Each solution I put about 30 shrimp, larva shrimp, and then I put it 24 hours. After that, I calculate how many shrimp are okay. Toxicity test with the SLC method use 5 kind of solution. So I mix another solution with concentration 100 ppm, 50 ppm, 20 ppm, and 10 ppm. Each solution I put about 30 shrimp, larva shrimp, and then I put it 24 hours. After that, I calculate how many shrimp are dead at each solution. And I was recorded, then the percentage of death was calculated. Okay, for GCMS analysis, I use the GCMS 7890 GC line types with a configure with an auto sampler and a 5975 mass selective detector. The mass spectrometer is operated in the electron impact ionization mode with a voltage of 70 electron volt. And for interpretation of GCMS mass spectrum, I using the Willy database and the existing National Institute of Standards and Technology or NIST on the Indonesian Herbs and Medicine Research Institute or Balitro database. Okay, this is the result of the SLC test method. I use 5 kind of concentration, 1000, 150, 20, and 10 ppm. Yeah, concentration of extract ethanol. And then each concentration I put 30 larva, and I left it about 1 day or 24 hours. After 24 hours, I calculate how many shrimp die. The experiment replication 3 times, so the number of die shrimp is average from 3 replication. For 1000 ppm from 30 larva, die about 11 shrimp, average 11 shrimp. 400 ppm about 9 shrimp. For 50 ppm about 7 shrimps die, and for 20 ppm and 10 ppm same, from 30 larva, each die about 5 shrimp. Okay, after I have data about the die shrimp from each concentration, and then I calculate the probability of LC. use with SPSS statistic prohibits. Yeah, this is the data. If probability about 0.3 or 30%, the LC about 543.423 ppm. For 0.35 or 35%, the concentration about 861.057 ppm. Yeah, and then for the red one, this is for LC50 or 50% of shrimp are dyed. The concentration of extract about 1741.059. So this number is LC50 for extract ethanol. Okay, this is the table of GCMS result. There are for enhanced fraction, there are 20 compounds with the high abundance are 5 compounds. Yeah, ester 13.79% abundance, and the second one ester 9.08%. The third one is pitol 8.54% abundance, and then linoleic acid, yeah, 8.71%, and for stigmasterol it's 7.23%, and another the abundance less than 4%. Okay, for ethyl acetate fraction, the result of GCMS, we found 19 compounds with the high abundance 33.25 for 2-oxatetracyclo, and then 14.9 is for linoleic acid. After that, the third one is 6.04 for hexadecanoic acid, same, yeah, acid, and then 3.68, this is for colex 5N3-ol 23 ethyl 3 beta 223 secondary, and then the last one, the high abundance is for 9%. Okay, we discuss about the result of the research. The first, for toxicity, yeah, the Cassia Ciamea.Lamk lips, the toxicity of LC50 extract 96%, about 1741.059 pm. Another researchers report that for LC50 of Cassia Ciamea.Lamk extract ethanol in rats, yeah, was found 9,600 milligram per kilogram body weight. It's reported by L.M. 2005. And toxicity of 70% ethanol extract of Cassia Ciamea.Lamk lips also reported by Wahyudi at 1999. He said that a concentration of 2040 milligram per kilogram body weight, it didn't cause toxic in white rat. Okay, according to Mayer BN 1982, an extract considered toxic if it has an LC50 under than 1000 ppm. So, for Cassia Ciamea.Lamk extract with the LC50 1741 ppm, it means not toxic. It can use for herbal medicine. Okay, how about the compounds that we found at Cassia Ciamea.Lamk extract with GCMS analysis method? Rakesh K. 2006 reported that gamma linoleic acid has potential anti-inflammatory, and Sen T et al. 2012 reported that stigmasterol has anti-proliferative PC3 human prostate cancer cell, and Clodary reported that 9,12,15-octadecantrionic acid methyl ester has activity anti-inflammatory, hypocholesterol, can prevent cancer, and hepatoprotective. Another researchers reported that hexadecanoic acid and vitamin A has antioxidant activity, and Pythal has antimicrobial, anti-diabetic, anti-cancer, anti-inflammation, and diuretic activity. It's reported by Peter, 2014. Well, the conclusion of the research are the first, for toxicity, based on the result, the LC50 of extra Cassia Ciamea.Lamk about 1741 ppm. It means the extract of Cassia Ciamea.Lamk not toxic for herbal medicine. The second one, according to GCMS analysis, there are many compounds of secondary bioactivity compounds we can found at extract of Cassia Ciamea.Lamk. So the Cassia Ciamea.Lamk slips is very potential to be used as herbal medicine. Well, for further research, I will be carried out on the anti-diabetes test using in vivo methods, as well as an acute or subchronic toxicity test using rat test animals. This research was funded through competitive grant project by the Directorate General of Higher Education, Ministry of Research, Technology and Higher Education of the Republic of Indonesia as a part of research grant to Binus University. So, for both of them, I say thank you for all support. Well, I think it's enough. I report about research result, and I hope it can use for another researchers for study about Cassia Ciamea.Lamk as a medicine herbal. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.